So thank you very much, Saye, for coming here today. And Saye is going to share his perspective on how innovative clinical trials can benefit patients in infectious diseases. Thank you.
Thank you. Thank you, Pavel. I hope I can work this, and it will be short, and you will get out your tea in time. So I'm a dumb clinician, and I apologize for that. I'm totally innumerate. I am totally out of my depth in this meeting. So I'm going to, so as I said to Jack earlier on, it seems to me that the trial design now can be so efficient that actually the risk of trial failure is because people are asking the wrong questions upstream. And I'm going to try and address that in the field, in my field, which is infectious diseases. So we're talking about innovative trials in relation to infectious diseases, and these are my declarations, which are also published online. And this doesn't work. So next slide, please. So in the pandemic, these are well known to you all. Okay, so that conventional drug development is not designed for a pandemic. Not only is it too slow, but it's not responsive to a changing virus, a changing epidemic, and a changing host immune response. So that's obvious. And in disease biology, people working in virology would have always told you that. They'd always told you that viruses are mathematically highly predictable. You look at HIV and hepatitis C, they're not like eukaryotes or the human immune system, which is more complex. So viruses effectively, think of a virus as an infectious chemical, which is bound by the laws of mathematics. So they do everything that you would expect them to do in the way that a chemical reaction would do. There are no options other than mathematical options for them. So the mutations will arise stochastically, they will enrich deterministically, you know that any virus entering a new host will adapt in the most parsimonious way. So it will always look for a way to become less virulent and more transmissible. Okay, so we know all that. Okay, and so there are things that we could do to better equip ourselves for the next pandemic. We need to look for good antiviral candidates, and one of the things that we saw was that there were plenty of compounds that would never have met the evidence criteria to be selected for a phase 3 trial in peacetime. We had to do it, but actually, once we disproved it, we wasted a lot of time disproving it even more. Research, and Richard will talk to you about that, was in a very challenging environment. I find in myself how easy it is to forget just how awful it was in the pandemic. You know, I was in Liverpool. I was rung one Saturday night by the duty manager saying, you know, we've run out of body bags. Now, that's not something that medicine ever trained me for. Okay, so facilities, staff, equipment, those challenging. Patients couldn't get to the hospital, couldn't use public transport. In the clinical research facility, we estimated that at any one time, 1 in 5 staff were off with COVID. And each study carries opportunity costs. So you put a patient into a crap study of a shit drug. Sorry, this is being that you're not studying a better candidate, and so bad trials cost lives. If you ever needed evidence that that's the case. So next slide, please. So what do we do for the next pandemic? Okay, the big one, the existential one. So like all of us here, we saw decision making. So by definition, you have to make difficult decisions with incomplete information. And everyone here, I think, has got an example of good and bad decisions made with the same incomplete data, right? So how do you make good and bad decisions? And it seems to me, and I'm going to really annoy every statistician here by saying that this process is in some ways inherently Bayesian. And you make decisions based on good or bad prior information, which you have almost always gained in peacetime. So this is what we have to do now. And specifically, how do we select candidates for the new disease X, better than we did before? How do we test them most efficiently? And then how do we, outside of the clinical trial that we do, prepare ourselves for actually treating patients in the event of an outbreak? So next slide, please. So we go on to repurposing, and I'm going to use repurposing in a very narrow sense. So I'm not talking about host-directed therapies, and I'm not talking about antivirals where you expand the antiviral indication. Okay, we're talking about repurposing largely off-target effects. And these were obviously the first thing that everybody reached for. And I would put to you, and I know many people disagree with this, that this is just a stopgap. All right. That if you, and you see these papers of highly potent antimalarial compounds, azithromycin, chloroquine, antidepressants, published as highly potent, but their efficacy in cell systems tends to be in micromolar rather than single or double-digit nanomolar, which is what I would call an antiviral. And there are common mistakes that we make in this area. We assume that there's a class mechanism. So we tried lopinavir, ritonavir, which was the first protease inhibitor for HIV, dirty drug, lots of side effects. And then we assumed that actually the best protease inhibitor, darunavir, would be even better. Well, actually, the first generation of drugs has got the most off-target effects and probably is the best one that you should go for repurposing. Don't assume that the licensed dose will suffice. And this is where you need to go and make some friends in the pharmacology world, I'm afraid. And so time and time again, I'll show you some examples of how we chose the wrong dose simply because it was a licensed dose. And the same is for the routes. I saw, for example, people talking about nebulizing some antimalarials to get into the lung at high concentrations. And you're giving a drug with a cardiac toxicity QT effect straight into the pulmonary circulation with no first pass effect. Any pharmacologist would be aghast at this suggestion, right? Of course, monoclonal antibodies are the first response, and there are some issues with them around scalability, around affordability, and around genetic barrier to resistance. So we've talked a little bit about that. Small molecules is probably where it's at, and I'll talk most of the time about this. And I won't talk about siRNA or gene therapy or any of the other things that we've come across. So next slide, please. So if you talk to people, you think the potency is the only thing that matters. But actually, there are three considerations in my mind, at least when you think about good antivirals. Okay, you think about a drug that's very potent, but you also need it to cover the breadth of natural variants in the population. And then you need a genetic barrier to resistance. That's to say that you don't just need one mutation to suddenly get a 100-fold change in your EC50. Okay, so these are three things that we need to consider. And if you think about, this is just a schematic, but think about where we are, monoclonals were highly, highly, highly potent when they worked. But against the natural breadth of COVID variants or SARS-CoV-2 variants, they were perhaps not so good, and they were easy to escape. So we tried to get around that by combining pairs of monoclonals. That slightly gave us a bit more genetic barrier, but not a huge amount, as it turns out. And where we need to be is in the high potent, high genetic barrier and broad breadth of coverage. And I think we are getting there. So for HIV, for hepatitis C, we've got that. And we've got that not because we have a single broad spectrum antiviral. That is potentially possible, but you know, and if you go to pandemic preparedness meetings, as I do sometimes, you find this term bandied around, and I find myself saying, well, name me one, right? And there aren't that many, okay? But what you can do is you can design broad spectrum regimens, and I'm going to talk a little bit about combination regimens in this context. So back to your basic virology. This is any old virus would look like that. There are some variants. Some viruses integrate in nucleus and genomic DNA, and some viruses primarily replicate in the cytoplasm, et cetera, et cetera. But virus, in the case of COVID, well, you can, let's just get the animation going. So monoclonals will bind onto free virions. They work in other ways as well, and they do stimulate the immune system. You can interfere with viral entry. Generally speaking, in all other viral diseases, these are fairly weak effects and easy to escape, and there are natural variants. You can interfere with nucleic acid replication, RDRP inhibitors, nucleoside analogs. You can induce hypermutation. So these are drugs that are do utilize RDRP to get integrated and then introduce catastrophic mutations. So molnupiravir, favipiravir, and ribavirin are three classic mutagens that are working in that regard. And then you get more specific for each virus, HIV, hepatitis C, will have protease inhibitors specifically directed against that virus. And with SARS-CoV-2, we've seen a number of those that have gone to success or bitten the dust. And then there are others that interfere with capsids and with virus release. Okay, so I'm going to talk a little bit about broad-spectrum antivirals when I come back to selecting a portfolio of drugs for the next pandemic. The other problem that we had as the pandemic evolved was that it became harder and harder and harder to show success. First of all, death and hospitalizations. That was about 7% when Paxlovid was tested, and it was 0.8% when molnupiravir was tested in panoramic. And this is not a complaint, it's a good thing, but it makes trial design quite challenging, and it also and in many of these trials, the inclusion of placebo arm is problematic. We could look then, if you don't look at death and hospitalizations, at symptom resolution, that introduces problems for open label trials. And then what tool do you use, which went, how, when, and this is open to use and abuse. And then you can use virology, much discredited in COVID, but critically important still, I think. Discredited because there's a disconnect between virology and clinical outcomes. And that there's a lot of technical and intellectual reasons why the virus from a throat swab might not be reflective of an underlying, might not be a good proxy for changes in a biological process. So here I've summarized some of the big small molecule trials. So in, I haven't got a pointer. Have I got a pointer here? Yeah, there you go. So nucleosides here, remdesivir, molnupiravir, and AT527, and then protease inhibitors down here. These are big pivotal trials, phase 2b and phase 3, and you can see some of the large numbers down here. And you can see that in some studies, which showed clinical benefit, there was a clear virological signal, and in some of them there wasn't. Okay, and there was no. And much of this relates to the way that you sample the virus. So the gold standard was a nasopharyngeal swab. How many people here have had a nasopharyngeal swab? Yeah. So I had one. It felt like someone was scraping the base of my brain, if done properly. And so you can't do it frequently, and that's a problem, okay, which we will talk about later on. So virological signals are not accepted by regulators for licensing of any new antivirals, but may be useful in early phase trials. In fact, I might just say that I think they are useful in early phase trials because apart from anything else, they show killing activity. They don't show saving activity, but they certainly confirm that what you set out to test can kill virus. So what about questionnaires? We've used this and we've tortured ourselves, silly Richard and I, on this. The first response was to adapt the FluPro questionnaire. But there are problems with it because FluPro questionnaire looks at multiple questions over multiple domains, and not all domains are informative for COVID. So if you look at loss of taste and smell, that's pretty good, but conjunctival symptoms are terrible. Yeah. And they don't all resolve in the same rate either. So your power, your precision, and then when you add these up into composite score, you're actually diluting a lot of the effects out as well. The early studies did use them and did find a difference based on them, monoclonal antibodies primarily. And then the FDA introduced a 14-point instrument, which has been used by multiple, which is probably the benchmark now. But the problem is that there's no standard way of, do you add up all 14? Do you look at them in classical respiratory symptoms, upper airway symptoms, systemic symptoms, as some people have done? Do you do it every day? Or do you just select the best ones that you like? So, for example, ensitrelvir selected 5, pre-selected for Omicron, they say, but we don't see any validation. A study called SPRINT was even worse. They looked at 14 over 5 days, then 14 over 3 days and got a bit more significance, then did 6 over 3 days and got a highly significant result. And as a result, they went on to a phase 3 study. Now, I think that's an approach that is slightly flawed, I think. In this room, people would agree. And panoramic, which is went the other way, just asked one question. Do you feel that you've completely got over COVID or not? And actually got a very clean result based on that one question. So different approaches and no standardization. Time to viral load negativity used as probably the main virological output of all these studies that in fact didn't show a difference. And there are problems with time to viral load negativity. You don't know whether the virus is dead or alive. And it's very dependent on which story you jump from, right? So if you, this is a slide I took off Nick White. So, if you jump from the 10th story or the 70th story, the result is probably the same. But the time taken is different. There are technical issues around swabbing as well. Who swabs you, and how much they dislike you, and how many cells they pick up. What's the volume? So swabs are not standard. The amount of cotton wool that you get on the end of a swab varies hugely, and we weigh them when we do our studies as a result of that. Okay, so there's a lot of technical issues as well around that. So this is a study done by the Oxford group and in their adaptive platform study, PlatCov, and they had some really interesting data, which basically shows, if I take you through this is a viral load by days of symptoms. So the viral load is changing depending on when you present. Yeah, you're probably at your most infectious at the start of your symptoms, and it goes downhill from there. The baseline viral load does vary by a calendar period, largely because of different circulating strains. So it is quite a difficult concept to get around. And one of the ways that we can get around this, of course, in a platform study, is that you're enrolling contemporaneously your controls and your subjects. But we can start to use model-based approaches to look at virological outcomes. And I'll talk a little bit about that. What can we learn from other diseases? So let's use the word prior wrongly in a different way. Let's use prior diseases and what we learned from HIV, hepatitis, and TB. So if you look at the pathogen burden, so this is the effect on treatment. When you start antiretroviral therapy, you get at least by exponential, possibly a 3-phase reduction in viral load. You see that with hepatitis C as well. And you certainly see that with tuberculosis. Now, the biological basis that underpins each of these is completely different. Yeah, and that's why I've taken these 3. And we need to understand a little bit about the biology of COVID and the next pandemic pathogen. And that's work that you can do in peacetime. All right. It suggests that there are multiple populations, but these populations could be genetically different. They could be, as they are with HIV, actually, because it's a very heterogeneous population. They can be anatomically different. So hepatitis C, they can be in the liver or free virions released in the blood, or in TB, indeed, within free bacilli or intramacrocyte bacillary or inside cavities, for example, or they can be metabolic differences. So there's some evidence that the slow phase of elimination is a population of bacteria that's metabolically different, uses much more lipid metabolism, has got lipid bodies when you stain it with Sudan red stains, et cetera, et cetera. So you have to tie what you see with what you think that is going on. And so with HIV, there are plenty of mechanistic models. There are variants of a fairly basic Bonhoeffer type model. You've got uninfected cells, infected population of cells, productively infected, etc., and then a clearance of cells and a clearance of virus. With HIV, there is an added dimension in that there are mature and immature cells, and this relates to integration of virus into host genomic material. And so what you see, which is the blue line, that the composite dynamics of viral eradication in HIV is actually made up of many different populations of of short-lived cells, long-lived cells, mature, immature, to give you this. And so when we introduced in 2018 a new drug called dolutegravir to replace WHO first-line efavirenz-based therapy, one of the reasons for doing it was that it was deemed to be more potent, but actually it just worked in a different way. And it got viral load down very, very, very quickly, which is great if you're a pregnant woman and your risk of transmission is highest at the time of delivery. So that was one of the studies that we were involved in. And let me just see. And so you can see that with the standard of care at the time, the rate of achieving an undetectable viral load was much faster with this new class action of drugs. So this is a little bit about tying in the biology with the clinical trial. With hepatitis, there was, this is a mechanism, a physiological model, which is semi-mechanistic, based on two mechanisms of viral, two mechanisms of drug action affecting the drug. One was to affect the drug release from the liver into the blood. So one is that the treatment prevents the secretion of virions into the blood. And the other one is that it prevents the infection new infection within hepatocytes. Yeah, and if you look at the model, you can switch off secretion and you get this line, or you can switch off a new infection and you get the red line, or you can integrate both of them and get this. And this, in fact, is akin to most of the observed data that you see. So with hepatitis C, you get a 2-3 log drop in the first 48 hours, because that's when the inflection point is, yeah? First 48 hours, you get this massive drop. That's largely due to the fact that the drugs stop the secretion of virions into plasma from infected hepatocytes. With TB, it's different. And so you've got what's called an early bactericidal activity of drugs and a sterilizing activity of drugs. And with TB, so antivirals mainly work on that first phase. With TB, you're mainly interested in the second phase because TB treatment is 6 months long. If you're going to get it down to about 4 months or 2 months or 1 month, then actually you need to have drugs with high sterilizing activity. And we know now from trials that rifampicin and pyrazinamide are big drivers of sterilizing activity, and isoniazid will drive the early bactericidal activity. The problem with TB drug selection is that everyone was using quick fixes and short-term experiments to select out drugs. So a lot of drugs, bedaquiline, which is one of the most exciting TB drugs discovered, actually was initially rejected because it had poor early bactericidal activity. So the questions, so you have to ask the right question, basically. All right. And you can see that if you use a model-based approach, and I'll get back to on topic again after this year, but if you have a population, average elimination here, and you use summary statistics, you will see that there is not necessarily always a good fit. But look at this patient and this patient. You know, they start at different. Initial pathogen burdens, but they actually eliminate all the same rate. And so if you use a non-linear mixed effects model, for example, you can get the residuals right down and you can improve your precision, and that changes fundamentally your trial design. Right, so let's get back on topic and look at COVID, SARS-CoV-2. So nasopharyngeal swabs are poorly tolerated. So patients, we're not going to thank you for doing them every day. In fact, they're not going to let you do them every day. So what we gain in sensitivity from an NP swab, we lose in precision of eliminating the slope. So isn't it better to have a less sensitive way, but to do more frequent sampling, okay? And that's the basis on which the Platkov team decided to do daily swabs from throat, and the agile method that we've used is a combined nose throat swab, and do it every day. And then track the viral elimination slopes, accepting that the sensitivity is diminished. Okay, using that approach, the Platkov group did see a difference in viral slopes. So they, first of all, they did show that in a large majority of people, there was a bi-exponential clearance of virus, that they integrated this into a model and they found a difference. And you can see here the clearance half-life of Paxlovid, a very potent drug, molnupiravir, a moderately potent drug, and against placebo. Right, now I'm slightly intruding into Richard's talk, which is the main course really. But we did a study of a combination molnupiravir and Paxlovid. And it was fundamentally a safety study to look at the safety of this combination in a dose de-escalation design. And there were not many patients enrolled into this. 16 patients received the combination against 8 controls. And if you look at the summary stats, there's no difference. Indeed, there's no possibility of any difference in that. But and Pavel will talk to this, I'm sure. But when you look at the effects, so we hypothesize that the major effect of any combination will be in the first phase of virological elimination. And indeed, there was a difference. And you can see down here, this parameter here, which gamma 1 is it estimates a treatment effect. Okay. So we're coming to the end. We're talking about preparation for phase 2 readiness for the next trial. And so you're all familiar with the 100-day mission. We need to get trials and vaccines out within 100 days, and diagnostics, obviously, within 100 days of any outbreak. There are inherent risks in assuming that industry will sort out the early phase to this. Industry, in fact, facing a huge disincentive. That there are market decisions that override public health concerns. Okay, that there is now a higher bar. If you're still looking for hospitalizations, well, there's a lot fewer of them. All right, and there are diminished incentives. We're not now treating everybody. We're treating high-risk patients. So the size of the market has gone down hugely, and as a result, many compounds are going into freezers for the wrong reason, just as they did for SARS-1 and for MERS, okay? So sotrovimab, for example, was developed for SARS-1, put in the freezer, and the precursor to Paxlovid was also developed and put in the freezer, okay? So these are major concerns. The biggest concern that I have also is that key populations are excluded. In the next pandemic, you're going to get ICUs full of people on ventilators, having dialysis and taking cancer drugs and immunosuppressants and steroids and all the rest of it. And these are people who would normally be excluded from any early phase trial, any phase 2 study. There are unrealistic expectations around broad spectrums. I've said my piece already. I'll carry on saying it, that there is always a trade-off between breadth and potency, but that you can circumvent that to a degree by thinking in terms of regimens. The problem here is that all drug companies that we have, bar none actually bar one, Richard, we can talk, all the drug companies we spoke, we said, we will first test our drug in monotherapy, and when it works, we'll test it in combinations. But pandemics don't work like that, you know. Marburg lasted 6 months, you know, Ebola, Sudan lasted 9 months, yeah. And so you can never in one pandemic test both, yeah. You have to test them side by side, yeah, okay, and that is the problem. So that brings me to my last point, which is that actually I think that more than ever now we do need publicly funded platforms. We need to incentivize it for the drug companies. We need to de-risk drug development, and of course, who's arguing for equitable access if it's not a publicly funded platform, right? So I'm going to finish very soon. I've said that twice already, I realize. Okay. So what is a broad-spectrum antibiotic? This is a virological sin, because what I'm doing is I am showing you cross-study comparisons, which you should never do, right? And the caveats here is that, so these are looking at EC50s and IC50s, but they vary by log orders depending on which cell you use, which variant of the cell you use, which virus you use, how much of the virus, what's the size of the inoculum. Do you inoculate first and then drug later, or the other way around, or at the same time? Yeah, okay. How long do you incubate? What's your readout? Is it a PCR readout, a plaque forming unit readout, or is it a reporter gene assay? How much protein is in your media? This is vast. 5% of fetal calf serum binds 96% of protein for lopinavir, okay? So your fraction goes from 100% to 4%, yeah, okay, of free drug. So these are big effects, but all those said, here's my stab at what a broad spectrum antivirals look like. Okay, and you can shoot me afterwards. You can see that flu drugs do what they do for flu and nothing else. Likewise, COVID. I've put in this corner here a common mutation that in neuraminidase, which is common in avian mutations as well. So some loss, but fortunately for us, thank God, highly pathogenic avian influenza, HPAI, is sensitive to, hasn't got this mutation, is sensitive to all drugs so far. Right, now if you look at COVID, there are variable protease inhibitors there are variable breadth. Some of these are COVID specific, some of these are pan coronavirus. And the two that you should watch out for is this one, ALG 558 and EDP 235, which have pan coronavirus activity. But if you look at the others, the broad spectrum ones, there are largely only two types. One are mutagens, which are modest in effect. And the other one are nucleoside analogs, and that's where we should be putting our money into, okay, remdesivir and its analogs. Right, I'm not going to go through what we're doing for time. I'm going to crack on now. What? 1 minute, I can do it easy. Okay, right. So we need antiviral libraries. These are the things we need. We need to know the viruses that it works against. We need to know what the PK looks like. We need to know whether it's safe. We need to know a little bit about drug interactions. And then there are other things that would be nice to have. And effectively, we can build model libraries. And we've started doing this in Liverpool. So for favipiravir, which you hear about, we have models of favi and its metabolite M1. Orally, intravenously, and in its intracellular metabolite FTP. And you can calibrate this to body weight. So we know the effect of body weight on light people and on heavy people. We can calibrate this to renal function, so renal impairment, CKD5 pre-dialysis. You can see, for example, that favipiravir doesn't change, but the metabolite increases, so potential toxicity from that area. You can look at the effect of loading dose to get an early viral kill. Okay, so you can model these out. And you can look at viral families, and you can see here we haven't adjusted for protein binding. You can see at standard doses of oral favi never had a chance to kill off SARS-CoV. So all those negative studies were still none the wiser. Was it because we didn't give enough, or was it because the drug doesn't work? Okay, so you do need that kind of data. We've done, say, for oseltamivir with a range of doses, with genetics in carboxylesterase mutations, renal impairment, and liver impairment. Okay. And then we need to manage drug interactions. So this was a German, Tom, German algorithm for prescribing Paxlovid, which I think looked a bit like this. Yeah. Okay. And so when it was when Paxlovid was licensed for use in the UK, the Scottish GPs, not one of them wanted to prescribe it. So it was left out to 4 Scottish pharmacists. We need to make this operationally deployable. We've developed a huge resource, the Liverpool Drug Interactions website. We return around 5 to 8 million searches every year across 220 countries. It's well used, it's in the guidelines, it's user friendly, and you can see the usual suspects, and we can tune our. It's got over 1,000 drug interactions in it, and then there's a whole pharmaceutic support around this. You know, can you give it renal impairment, liver impairment? Can you give it to pregnant women? What if your patients can't swallow? Can you crush the tablets? Are they available in liquid? Can you give them with NG tubes, etc.? So I'm going to finish here because I'm sorry I've taken so long. By saying clearly we do need better selection upstream of a trial design, and we need to start building these libraries in peacetime. And many thanks to the rest of the group. Thank you.